I'm Rachel Armstrong, Professor of Experimental Architecture at Newcastle University and Coordinator for the FET Open Living Architecture project, which ran from April 2016 to September 2019. We're a 6-partner strong consortium, including Newcastle University, the University of the West of England, University of Trento, Spanish National Research Council, Liquefer Systems Group, and Explorer Biotech. Living architecture is a modular bioreactor wall that processes waste. It aims to change building impacts, which are a worldwide challenge and account for around 40% of our carbon footprint, which is a bit more than transport. Living architecture achieves this by altering the effect of our domestic lifestyles on the environment, where constituent households do not just consume resources, but also generate housework, transforming the notion of waste. Right now, the best we can do in terms of energy consumption is net zero impact. If we include on-site renewable energy sources, we might even put some energy by or trade with it on the power grid. Energy use, though, is only one aspect of our lifestyles, and net zero solutions do not address the broader issue of resource consumption. Whatever way we produce energy, if we continue to consume at our current rate, then we'll keep on impacting the environment negatively, as nothing will temper our consumptive lifestyle patterns. By applying 21st century biotechnical insights to regulate the robust processing power of microbes, living architecture enables us to give something back from our waste, which is turned into usable resource again. Specifically, it produces energy in the form of bioelectricity, as well as cleaning water, removing waste, neutralizing pollution, reclaiming phosphate from washing liquids, and producing particular forms of biomass, all of which can be circulated within the home, as well as being good for the environment. Living architecture is so much more than a multi-utility system. It is the foundation for a circular economy of the household. The key question that living architecture therefore raises is just how far can we design and direct the microbial processes within waste to perform housework? Our consortium set out to prove this principle by bringing together the insights of metabolic networks, synthetic biology, genetics, bioenergy, bioelectronics, informatics, and design to produce a freestanding interior partition wall about the size of a large bookcase, suitable for installation in a bathroom or kitchen. The wall is made up of bricks. These double up as integrated building blocks, where each brick is also a next-generation selectively programmable bioreactor and ideal home for microbes. 3 different kinds of bricks are used in the wall: the microbial fuel cell, an algae photobioreactor, and a synthetic consortium bioprocessor. Each type supports different kinds of microbes, and together they are assembled as modularly stacking, standardized building segments. Liquid domestic waste, in the form of urine and grey water, is used as a feedstock, which is sequentially passed through living architecture's various chambers. As a living structure, the wall's operations can be likened to the workings of a cow's stomach. Each brick differentially processes liquid and organic matter within its gut, changing the substrates they receive into a range of metabolites that are moved on to the next chamber, where further transformations take place, and so on. Each brick can be assigned a particular task, which is possible by altering the microbial populations and then sequencing them spatially. In other words, the system is programmable and can be thought of as a metabolic app. 2 basic types of metabolism are used in living architecture. One is anaerobic, based on a technologically mediated composting process that can be harvested for electricity, water, and oxygen, As a standalone unit, it's known as a microbial fuel cell, which is a kind of battery that's powered by organic waste. The microbial fuel cell consists of an anode, selective membrane, and cathode. The hardware captures electrons from the bioelectrical activity of the biofilm that forms on the membrane to provide small amounts of usable electricity. At the same time, the fuel cell cleans water and produces other metabolites. The system is self-powered, and its actions are coordinated by an AI, which detects the amount of electricity produced by each brick and regulates the inputs accordingly to optimize the system outputs. The other metabolism is photosynthesis, which is carried out by tiny green organisms, or algae, in a photobioreactor, which traps sunlight and carbon dioxide that is used to produce solid biomass. Our system is gravity-fed, with a unique geometry and triple baffle system that promotes mixing. The main challenge of the project is to bring the different systems together so their various products can be meaningfully exchanged. We achieved this by boosting the electrical power of the microbial fuel cells by introducing oxygen from the photobioreactors into the cathode as a powerful electron acceptor. Achieving this integratability changes the very notion of waste, because the outputs of one system can be used directly by another, generating a cyclical economy of matter powered by metabolism. The final bioprocessor type that we developed is a synthetic bioprocessor, which examined the biological flexibility of the metabolic apps, helping us establish just how far we can design the actual metabolic reactions using synthetic biology techniques. While our modified system cleaned nitrous gases from the air, our indicator product was organic phosphate that was reclaimed from detergents. Inspired by the configuration of the microbial fuel cell, the synthetic bioprocessor was made up of a farm and labour module, separated by a selectively permeable membrane. The farm system feeds the labour module with a rich supply of carbon from organisms that overproduce sugar. This passes through the membrane into the labour module and reaches workhorse organisms whose genes are well known. The sugar-rich environment enables the design of new metabolisms, as under these conditions, enabling us to mix and match genetic sequences as a mixed population of modified microbes forms in ways that are not possible in nature. Specifically, these new metabolisms can reclaim phosphate from detergents and remove polluting nitrous gases from the air. The ability to stitch different metabolic building blocks together between microbial species greatly increases the metabolic range for design and enables us to work with these processes at scale. During the project, we created 2 new genes, one with a patent granted and one pending. We also developed a new software called Dulix, which is a web-based DNA library design tool that makes gene sequencing more intuitive. The new knowledge produced in this project also secured further funding in new grant applications for environmental remediation and architecture, multiplying the revenue generated to around 10 times that of the original €3.2 million investment. Throughout the project, we also considered the social acceptability of the technology through various brick prototypes that were exhibited in biennales and international exhibitions. The first prototype was a simple hack of a brick, turning it into a microbial fuel cell, which brought together structure and process. This was displayed at the Building Centre in London in 2017 and during the Venice Architecture Biennale in 2017. We also made structures that could simultaneously host unlike environments, such as this brick designed for the 4th Tallinn Architecture Biennale in 2017 that links together photosynthetic and anaerobic organisms, enabling them to exchange metabolites with each other. Our final brick prototype, based on this principle, contained 4 chambers separated by ceramic plates, where we reclaimed 100% of the phosphate that was introduced into the system. And this diagram shows how the spatial sequencing of the Living Architecture project was configured, thereby completing the metabolic app. The final wall shown here used synthetic organisms, so it was explored in a laboratory rather than a social context. Although we couldn't use the original Living Architecture wall in a public setting, we generated a prototype system that was shown at the Whitechapel Gallery in collaboration with artist Cecile B. Evans as part of the Is This Tomorrow exhibition. The installation is an apartment space where a screen system is powered by wild-type organisms within a set of microbial fuel cell bricks. The work can be thought of as a post-human household, inhabited by ghosts of the past, present, and future. The uptake of living architecture raises important questions about how we live. For starters, we must question the need for a 230-volt electrical main supply within our homes. Given that we step down a lot of our electrical power, the prospect of 12-volt lifestyles is raised, an amount that could be reasonably provided by the living architecture infrastructure. Stepping down our power supplies does not mean a less advanced lifestyle, but one that is experienced differently. In the video, we demonstrate that microbial fuel cells in the living architecture system can power a window-opening robot. Rethinking how we perform housework opens up new frontiers in low-power electronics and robotics and invites an era of innovation in low-power domestic appliances, such as using ultrasound to clean our clothes or employing thermocaloric materials in refrigeration. Tim Garrett, an atmospheric scientist, Matteo Scriselli, a mathematician, and Stephen Keen, an economist, have recently looked at the economy through a thermodynamic lens. They concluded that although current efforts to reduce greenhouse gas emissions are focused on improved energy efficiency, this has the side effect of making it easier for civilization to grow and consume more. They suggested the way forwards is keeping the economy at a steady state, where the energy we use is devoted to maintaining our civilization and not expanding it. Additionally, previous fossil fuel-based innovations must be phased out while innovating new non-fossil fuel technologies. A platform for a circular domestic economy that enables us to reconfigure our homes and cities, living architecture can help achieve the European Green Deal with a new thermoeconomics as the foundations for a regenerative society. Forming the basis of an economy that works for people, its operations can be customized and implemented in ways that promote the European way of life, which includes fairness for the natural world. To build an uptake community, we are developing a digitally enabled version of a brick to increase the usability of living architecture. This active living infrastructure controlled environment, or ALICE prototype, is supported by an EU Innovation Fund and provides a relatable digital experience that is powered by waste and communicates how microbes can perform valuable actions within our homes using real-time energy data to produce animations and online feedback loops that reinforce how we can help them do this. Supporting the EU's digital strategy, this ambitious technology embodies a new generation of digital devices that do not rely on fossil fuels, equating smart technology with environmental remediation. These breakthroughs would not have happened without the FET funding scheme. Securing 100% finance for risky, innovative projects is essential for bringing about step changes in innovation needed for genuine disruption. The whole experience has been a fantastic training ground for building an ambitious vision, organising complex projects, identifying shared needs, managing budgets, and benefiting from industry expertise. The project-based approach has enabled the consortium to come together around a shared vision. Despite our different disciplines, skills, and backgrounds, and make it come true. An unexpected further avenue for applications of living architecture arose during the COVID-19 pandemic. Spotlighting the presence of microbes all around us, even in our homes, the recent lockdown confined us to indoor spaces, where even before the crisis, we usually spend around 90% of our time. Now that we're spending even more of our time indoors, we can think beyond the biology of the virus and into the complex systems that shape the spread of the pandemic. Specifically, we can consider how transmission chains can be interrupted within our domestic environments, where we are most likely to contract the virus, by regulating the overall microbial health of indoor spaces through the installation of living architecture. Currently, cities like New Haven, Connecticut, are using sewers as COVID-19 early warning systems, or canaries in the coal mine, to track the levels of the novel coronavirus circulating inside their populations. If someone is affected with the virus, it shows up in their feces even before they may feel sick. Virus-flecked feces make their way through sewage systems, and checking in on all that sewage gives public health officials another layer of data on the extent of the outbreak. Fed with waste from our bodies, our natural microbiota and any pathogens will enter the living architecture bioprocessor system as we use it. A particularly interesting observation, which has been explored since an early stage in the development of the Living Architecture project, specifically in relationship to removing synthetic organisms, is the ability of the microbial fuel cells to kill disease-causing pathogens in their feedstock. Demonstrated by the University of the West of England, who is currently looking for COVID-19 samples to trial along with hepatitis B and other pathogens, early findings are not only a novel breakthrough for the sanitation sector, but also creates opportunities for regulating the microbiome of the built environment, like an external immune system. So at some point, our microbiota will reach the microbial fuel cells, where through microbial action, they are either incorporated into the anaerobic biofilm or removed from the system. Once a person's or community's microbiota and biomolecules have entered the living architecture system, they can be used for health monitoring more generally. With its integrated systems of artificial intelligence, sensors, microbial actors, and robotic actuators, living architecture provides a programmable infrastructure that can help us rethink how we manage our individual and collective health within indoor spaces to attenuate, for example, outbreaks of infectious disease. Providing a platform for the early detection, localization, and even removal of pathogenic agents from the indoor environment, a near future scenario within a building is through the installation of living architecture. On the detection of pathogens in the system, artificial intelligence and robotic actuators will activate air conditioners and operate an early warning system that, depending on the organism and context, acts according to an institutionally determined protocol. While living architecture won't solve the immediate COVID-19 crisis in its current form, as COVID-19 has rarely been found in urine, it does alert us to the near future role of the productive actions of microbes within the home environment and even workplaces that enable new forms of economy, health, medicine, public health, and disease prevention, all while making clean energy. Of course, these latter scenarios are speculative right now, but future outbreaks of infectious disease will worsen with a climate emergency and will not be won through an exclusively biological approach that only focuses on a specific organism. As we have already witnessed, our behaviours are also critical in our health and well-being. Living architecture helps us rethink how we make better use of our living spaces, where we spend so much of our time. And even in its current proof of principle form, this combined utilities platform is an important innovation space. A foundational platform and a way of thinking about our relationships with microbes and resource consumption at a critical time in our history, living architecture genuinely offers an alternative approach than resource consumption and industrial fossil fuel-based systems. It can even change our values, our environmental impacts, and economies. Working with live organisms as an extension of their existing co-inhabitation with us creates the possibility of a new relationship with nature that is not a panacea, but a different way of acting, thinking about, and valuing our world. Not all roads to innovation are direct, efficient, or linear. Not all outcomes are immediately profitable with respect to the dominant economic model, but innovation itself can be empowering and help us combat challenges in new ways. Something occurs during an ambitious FET project, something connected to perhaps the most important things that ambitious collaborations can offer: a commitment to shaping an uncertain future, an investment in those who will be shaping that future, and the formation of community through an expression of trust and sharing that makes all the difference.